皆様、大変お待たせいたしました。これより株式会社ベリタスによるウェブ講習会を開始させていただきます。本日はリキッドバイオプシーをトゥモローと題しまして、ストレック社より講演いただきます。コンテンツに関しましては、現状と展望についてっていうところで、40分から45分のプレゼンテーションの後、10分ほどの質疑応答の時間を取らせていただきます。本日に関しましては、ストレックの研究開発の部門より、ニコラス・ジョージ博士、また、ジン・ジー博士にご講演いただきます。ストレック社は独自の細胞治療技術を用いまして、リキッドバイオプシーの検体の保存というところで貢献している会社になります。それでは開始させていただきます。Professor Nicholas George and Jing Ji, please start your presentation.
Okay, can you hear us? Thank you all for attending this webinar. I'm going to provide a brief agenda for today. So before we can walk you through how liquid biopsy may look tomorrow, we'd like to walk you through what has led up to this point. So initially, we'll provide a background on Streck, and then creation of the Streck stabilization portfolio with emphasis placed on our blood collection tubes and urine preserve. And then we'll talk about the current interest in liquid biopsy, and this stems from non-invasive prenatal testing, which is a market that's currently saturated, and then newer markets that are just now expanding, and there's a lot of room for growth, and that is liquid biopsy using blood or urine. And then we'll talk about later technologies also. And this will be followed by a question and answer session. So a little bit of information about Streck. Streck develops and delivers quality control and diagnostic products that touch millions of lives, that help validate the reputations of laboratories, hospitals, and clinicians all over the world. So Streck was we're the first developers of multiple technologies. Our core technology is cell stabilization. And related to this, our first hematology control, which was actually a platelet control, was introduced by Streck way back in 1979. So currently, Streck is a global leader in the stabilization technology portfolios. We hold over 98 patents in the United States, Canada, Australia, and Europe. And then we're also the biggest supplier of stabilization tubes currently in the world, with millions upon millions of blood collection tubes sold yearly. So this is just a brief outline of the stabilization portfolio offered by Streck. So I'll talk about a few of these, but they include the cell-free DNA blood collection tube, the Cytochex blood collection tube, RNA Complete, and then two other preservatives, one meant for urine and the other meant for cell or tissue suspensions. So there's multiple areas of interest for liquid biopsy. So while most people focus on oncology, and that'll be the topic today, there are multiple other areas. And the first actually stemmed from reproductive care. Liquid biopsy or non-invasive prenatal testing in reproductive care is now the standard line of testing, where the clinician only needs a 10 mil sample to diagnose chromosomal translocations and diseases that result from that, such as Down syndrome. Liquid biopsy is also liquid biopsy assays are also being developed in areas of cardiology as well as organ transplantation. And then both Jing and I will touch on the oncology area as we go forward in this talk. So uses in oncology, there's multiple uses of liquid biopsy. So this can include detection and screening for cancer, diagnosis, prognosis, and prediction, and then also therapeutic response and minimal residual disease detection. And I'll talk a little bit more about these in subsequent slides. So what analytes are we interested in a collected blood sample for liquid biopsy? So you go to the middle of this diagram, you see that circulating tumor DNA, circulating tumor cells, EVs, which are extracellular vesicles, and then cell-free RNA or circulating tumor RNA are four of the most common analytes that are tested for. And our stabilization tubes specifically target each of these analytes. And we'll talk about that in subsequent slides. So this slide is mostly related to assays that are either currently offered in the United States or are up and coming in the United States. So there's a whole bunch of different oncology tests that are based on liquid biopsy, and these can be either a single cancer indication, like a single gene test, or they can be for a multi-cancer indication, such as the Foundation One and Guardant Health NGS-based assays, where you're looking at anywhere from 73 up to 324 different genes at once. Some of these are approved in the United States by the Food and Drug Administration, and those are the ones listed with yes on the right. So these can include the EGFR mutation test offered by Roche, and others, for example, the epigenomics test, which is approved for colorectal cancer screening, and this looks at a single methylated gene. And then again, Foundation One and Guardant Health look at 73 up to hundreds of genes using NGS-based technology, and I'll talk about the Guardant Health assay in just a minute. So liquid biopsy is currently used in the clinic, and some of these assays are FDA approved. So why are we interested in liquid biopsy? Well, liquid biopsy offers multiple advantages over a regular or a traditional tissue-based biopsy. So as the name implies, these are non-invasive. All you need to do is draw the blood sample from the patient. Tumor tissue is not actually required. Liquid biopsies are rapid, they're precise depending on the assay, they're real time, and then most importantly, they can be repetitive. You're not having to isolate a tumor tissue from the patient of interest multiple times, you just draw their blood. So this allows for repetitive and longitudinal based studies to monitor cancer progression and recurrence. So what's the overall workflow for liquid biopsy? So liquid biopsy is dependent on accurate analysis of draw time samples. So EDTA tubes, for example, you need to analyze out of an EDTA tube within 4 hours of draw, as per most protocols. And the problem with that is blood is prone to deterioration with storage time. And an example of that are white cells or white blood cells in the collected blood sample. As the blood sample sits at room temperature, the white blood cells degrade, undergoing apoptosis, and this leads to a release of apoptotic DNA into the plasma, which then dilutes the cell-free DNA concentration with white cell specific DNA that's not useful. So it's not representative of the true liquid biopsy. And then on the right, it's just a quick diagram of how liquid biopsy is generally used. So blood is collected, plasma is isolated, and then from that plasma sample, nucleic acids, be it cell-free DNA or cell-free RNA, or even extracellular vesicles, are isolated and then utilized in downstream methods. So why is liquid biopsy important, or why is all this important? So a few definitions. So the total population of circulating DNA in plasma is referred to as cell-free DNA. That amount of cell-free DNA that's derived from tumors is the circulating tumor DNA fraction, or the ctDNA fraction. And then in normal healthy individuals, your cell-free DNA concentration ranges from anywhere from 0 to 100 nanograms per milliliter plasma in a healthy individual. And then here at Streck, with most of our donor samples that we work with using generic QIAGEN-based extraction techniques, we usually see healthy patients ranging from 2 nanograms up to about 20 nanograms per mil plasma. What happens in cancer patients? Well, depending on the cancer, some cancers are higher shedding, releasing more circulating tumor DNA than others. So in some of these cancers, cell-free DNA concentrations can actually be almost up to 1,000 nanograms per milliliter. And then importantly, the fraction of circulating tumor DNA in cancer patients, whereas in some patients, this can be almost 90% of the total circulating cell-free DNA. In other patients, this could only be 0.01%. So it is like trying to find a needle in a haystack with these patients that have the lower concentrations of cell-free or of circulating tumor DNA as a percentage of their cell-free DNA. So low mutant allele frequency is what, or mutant allele frequency is what most people look at in terms of circulating tumor DNA. So the low mutant allele frequency in many cancers coupled with treatment or surgical resection where you're removing the tumor makes identifying these mutants extremely difficult. So it is like trying to find a needle in a haystack analogy. So two different techniques. So targeted NGS liquid biopsy assays. So these can get down to about 0.2% allelic frequency as a total, whereas digital PCR, like droplet digital PCR, this can get down to even greater allelic frequencies. So if you have a patient that has a low starting concentration of circulating tumor DNA and you want to determine whether or not they have mutations, sample stabilization is critical. And this is because as white cells break down, that white cell DNA contaminates the draw time concentration of cell-free DNA and makes identifying mutant alleles that much more difficult. So sample stabilization is critical. So that's where Streck fits in. So cold chain free transport. So our blood collection tubes, for example, the cell-free DNA blood collection tube shown here, allows for transport of blood samples through the mail at room temperature and then processing 1, 2, 3, 7 days later, instead of having to process within that short 4-hour period that we have to use for EDTA tubes. So next, I'm going to walk you through a few of our offerings for the blood collection tubes. So the one that I've already talked about is our cell-free DNA blood collection tube. So how this works is it stabilizes nucleated blood cells, and it specifically white blood cells, and that minimizes release of genomic DNA as a function of storage time. So in this tube, cell-free DNA is, or the draw time concentration of cell-free DNA is stable for up to 14 days at room temperature. And then also in our IFU, we've also provided data that circulating tumor cells are stable in this tube for up to 7 days at room temperature storage. So as I talked about earlier, the Guardant 360 assay, which looks at roughly 73 different genes at once in the liquid biopsy using next generation sequencing-based methodologies. The Guardant 360 assay was recently approved by the US Food and Drug Association for lung cancer as an initial indication. So liquid biopsy is no longer just research use only method. It is FDA approved for monitoring lung cancer patients using the Guardant assay. And the Streck cell-free DNA blood collection tube was the tube that was approved with the Guardant 360 assay. So anybody undergoing assay with the Guardant 360 assay is drawn into our Streck cell-free DNA blood collection tubes and then processed at later time points in their laboratories. So what about other plasma nucleic acids, such as cell-free RNA? Well, cell-free RNA, unlike DNA, is an unstable molecule. So the majority of cell-free RNA that's found in blood plasma is actually that proportion that's encapsulated within extracellular vesicles. So cell-free RNA is assumed unstable and thus protected by packaging within these extracellular vesicles. EVs fall into are made up of a lot of different types of vesicles, and these are all defined based on size. So the most common are the exosomes, or the most well-known are the exosomes, and these range from anywhere from 50 to 100 nanometers. And then the second most common are microvesicles, and these range from roughly 100 nanometers in diameter up to 1,000 nanometers in diameter. So cell-free RNA is packaged within these EVs and protected from degradation in the blood sample. But the problem here is all cells in a collected blood sample, be it red blood cells, white cells, and even platelets, release extracellular vesicles as a function of storage time. So if you want to maintain draw time concentrations of cell-free RNA, you have to prevent release of extracellular vesicles from cells as they deteriorate in the collected blood sample. And I'll talk about one of our tubes intended for that in just a minute. Okay, so why is cell-free RNA important? Well, that's one of the next interesting analytes on the horizon for liquid biopsy. So plasma cell-free RNA analysis can be used to monitor health, pregnancy, and disease status. And this is just an abstract that I pulled from a recent Nature article where these authors are utilizing cell-free RNA analysis using global expression analysis to reveal patterns of normal pregnancy progression, as well as the risk of developing preeclampsia prior to its occurrence. So you can utilize the transcriptome through cell-free RNA analysis to monitor disease status, similar to what's currently done with mutation analysis with cell-free DNA. And this is just one example. So which product do we manufacture for cell-free RNA analysis? So that's our newer tube. It's the RNA Complete BCT. So the RNA Complete BCT maintains the draw time concentration of cell-free RNA and extracellular vesicles, such as exosomes, for up to 7 days when stored at room temperature. So in addition to stabilizing those white cells, we're also stabilizing other cells in the collected blood samples, specifically red cells and to a lesser extent platelets. Immature red cells, also known as reticulocytes, are actually marked by their intracellular presence of RNA. So as retics degrade, they release RNA also. So there's multiple sources of potential RNA contamination in a collected blood sample. So again, stabilization is key for monitoring those critical draw time concentrations of cell-free RNA. So next, I'm just going to present a few slides on our RNA Complete BCT product. So the first thing we looked at with this tube, if we wanted to maintain draw time concentrations of cell-free RNA, we knew that we had to maintain draw time concentrations of extracellular vesicles because these encapsulate all of the cell-free RNA. So in our hands, we use a pretty rough method of determining concentration of extracellular vesicles, and that is using the Malvern nanosite. So using Brownian motion, this just gives you a glimpse of extracellular vesicle concentration in plasma samples. And as you can see in the upper right-hand panel, fold change in EVs in a normal EDTA tube from anywhere from 4 hours out to 8 days. There's over 100-fold change in extracellular vesicle concentration as a function of 8-day storage time. In the RNA Complete tube, this is maintained to near draw time levels. So we assume that by stabilizing draw time extracellular vesicle concentration, we should also be maintaining cell-free, by maintaining So by maintaining draw time concentrations of extracellular vesicles, we hypothesized we'd be maintaining draw time concentrations of cell-free RNA also. And that's indeed what we see. So RNA Complete BCT maintains cell-free RNA out to 7 days also when the blood sample is stored at room temperature. And here we're just looking at, on the upper right-hand panel, we're looking at bioanalyzer trace. So what you see on day 7 in the EDTA tube is an increase in RNA. In the Streck RNA Complete tube, you do not see that increase. And in the bottom panel, we are using droplet digital PCR for a generic housekeeping gene, ubiquitin. And as you can see in the EDTA day 7 samples, there's a dramatic increase in the abundance of ubiquitin B, whereas the RNA Complete tube maintains roughly draw time concentrations of this. cell-free cell-free RNA concentration is maintained in the RNA complete BCT out to 7 days. And then we also have a tech note on our website demonstrating use of the RNA complete tube with RNA-seq. So here we're taking an unbiased approach and depleting out ribosomal RNAs and then utilizing the remaining RNA for gene expression analysis. And when compared to the EDTA tubes, the RNA complete tube, we're still able to identify roughly 12,000 genes. Those that change expression significantly as a function of storage time. In the EDTA tubes, you see almost 1,500 genes change in statistically significant concentrations. In the RNA complete tube, this is much closer to draw time, where we only see roughly 10 to 20 genes change in abundance. And a lot of these are really low expressing genes, so they're probably less important. So the workflow for our RNA-seq data and how this can be repeated is available on our website. So to conclude, for the blood-based liquid biopsy section, based on ease of blood sample acquisition versus invasive traditional biopsies and breadth of analytes available, such as cell-free DNA, extracellular vesicles, cell-free RNA, and circulating tumor cells, blood-based liquid biopsy is fast becoming routine in the clinical setting. This is already realized for non-invasive prenatal testing, which is basically saturated, and is now becoming realistic in the realm of oncology. And the Garden 360 assay is a perfect example of that, for which our cell-free DNA tube was approved by the US Food and Drug Association. Maintaining precious blood sample stability is a must. So we offer two different options for that. For cell-free DNA work, we offer the Streck cell-free DNA blood collection tube, and then for cell-free RNA analysis and extracellular vesicle work, we offer the Streck RNA complete BCT. And if you are considering including a Streck BCT in assay development, please contact your distributor, and they can get in touch with us, and we can provide important insight on what you need to include on developing a assay with our blood collection tubes. So with that, I turn over the urine portion to Jing.
Hello. So now let's switch gears from blood and talk about urine. So urine is a very unique biofluid in a way that you can find systemic biomarkers that are secreted in the urine. And then you can also find biomarkers that are directly derived from urinary tract or urological systems like prostate, bladder, or kidney. So for the analytes of interest, you can find in urine that include metabolites. Most of them are small molecules, cell-free DNA and proteins, and cell-free RNAs. That includes all sorts of RNAs like messenger RNA or microRNA. And those are mostly found encapsulated in the extracellular vesicles or exosomes. And then in the exosomes, you can also find exosomal DNAs or exosomal proteins. In the cancer research field, people are particularly interested in exfoliated cancer cells that shed into the urine, for example, bladder or prostate. And then for the infectious disease field, a lot of people or clinicians are interested in looking into bacteria. Either they look at pathological bacteria that cause diseases like UTI, urinary tract infections, or researchers that look at microbiome. And then for the viruses, some viruses, for example, like Zika virus, that they can find in urine as well. And there are actually available tests for Zika virus on the market. So for the downstream analysis, cytology is used a lot for cancer detection and diagnosis, and then also all sorts of DNA and RNA PCRs and sequencing. For the protein work, LC-MS is often used, and also I didn't include here are immunoassays, which is mostly ELISA-based, and then also the most recent single-cell technologies as well. So there are quite few urinary liquid biopsy tests on the market. Some of them are approved by FDA already, and some of them are still seeking approval. So these are mostly focusing on prostate cancer and bladder cancer. And if you look at the target, so that basically cover all the targets I just talked about, all kinds of DNAs and RNA, DNA methylation, and also proteins and cells and cell surface markers in particular. And then all the technologies, including all the PCRs, microRNA microarrays, and then a lot of immunostainings for the cell surface markers, and then NGS and methylation-based PCRs are of interest as well. So there are a couple of strengths of using urine for liquid biopsy when you compare that with blood. So urine is considered a truly non-invasive collection, and the collection is fairly easy. It can be done by anybody. So the patient doesn't need to go to a lab or a clinic or hospital to get it done. So they can just do it at home if they have the collection device. And then the large quantities of urine is usually available when you compare with, 10 ml blood draw or 20, if you do multiple tubes of blood. And the urine matrix is considered less complex than blood because a lot fewer cells and then fewer protein levels, unless you have kidney dysfunction. And urine gives actually important information from the cells shedding directly into the urine. At the same time, it also reflects biomarkers that are transporting from circulation as well. And then the other cool thing is the multiple urine biochemicals such as glucose, ketones, bilirubin, and total protein. They already have very well established clinical assays to determine the healthy and pathological states of the patient. So that can help with the diagnosis process. But what about limitations? So the concentration of certain markers in urine can be quite low, and then they can vary upon different collection time during the day and also depend on the hydration level. So because of this reason, the normalization can be challenging. Some people use creatinine as a normalization method, but there's no standardization on the normalization part. Also, for certain biomarkers, for some patient populations, the dynamic range of those markers can be quite wide. The other thing is urine is actually a pretty harsh environment. So the high urine salt is not helping with your LC-MS analysis that can mess up the analysis and processing. The urine crystals actually happen quite often, especially in the older patient populations. And that can actually co-precipitate during centrifugation with other analytes of interest, for example, cells or bacteria, and that can further introduce interference of your analysis. And then also in patients, there's quite a wide range of pH. So we consider blood, compared with blood, that's pretty consistent, but in patients, in the urine samples in patients, sometimes it can vary from 2 to 10. And then if you're interested in studying cell-free DNA, then the urine has actually very high level of DNAs that can degrade DNAs very quickly. So urine actually has 100-fold higher DNAs level when compared with the level of DNAs in the blood. And then the other thing to keep in mind is the urine is not sterile. So microbial growth actually causes the degradation of your analytes and introduces other interference as well. And last but not least, similar with blood, there are cells in the urine and they lyse and they also release genomic targets that interference with your analysis and your target of interest. And then, so this slide actually shows the whole process from urine collection to downstream analysis. So usually you go to hospitals or clinic or a lab to get the urine collected in the cup. And then sometimes they require clean catch or first morning void to kind of have a better concentration of a lot of the analytes you're interested in. And then if they want large quantities or more consistent data, they sometimes require 24-hour collection. And then after collection, if no preservative is added, so these urine samples will be stored at cold temperature or refrigerated temperature or at frozen, so before they get shipped. And then during the shipping, there also needs to be refrigerated or using ice pack or ice box, or they're shipping frozen as well. And then after they arrive at a testing facility, depends on what you're looking at. So you can either look at whole urine for biochemical analysis or metabolomics or proteomics study, or an easy centrifugation step is needed. If you're interested in like bacteria or cells or cellular component, you will take the sediment. And if you're looking at cell-free DNA or RNA or proteins or viruses, then you will take the supernatant. If you're especially interested in extracellular vesicles or exosomes, another EV isolation step will be needed. Either ultracentrifugation and precipitation or any isolation columns. Those are the three most commonly used method for EVs. So Streck actually has a urine preserve product that can help eliminate the cold chain transport and storage. This is, so the urine preserve is in a single-use container that can be used to preserve 25 ml to 100 ml urine. And then, so this can be easily used by anybody, including the patients. And then it contains a signature PCR compatible blue dye. And then they will provide 7-day sample stability when you store at room temperature. So this will allow plenty of time for shipping or storage before it arrives at the testing facility. And after that, you can continue with your downstream assays. So the next few slides, I'm just going to share with you 3 studies that we have conducted for different analytes. So this slide, the first study I'm going to share with you is for cell-free DNA. So we collected first voided morning urine from healthy donors, and we treat them with Streck urine preserve at different ratios and store at room temperature for up to 7 days, and we compare it with the unpreserved sample in parallel. And then we use the QIAGEN's circulating nucleic acid kit with modified protocol to extract urine cell-free DNA. And we use ddPCR to quantify an oncogene, KRAS, in the urine. So as you can see in the figure, in unpreserved sample, at day 4, almost none of the KRAS copies can be found. And then at the with the urine preserved sample, whichever ratio that you're using, either from 1 to 10 or 1 to 30. So that kind of reflects on the 25 to 100 ml urine that it applies on. So you can, all of the samples they maintain at the draw time. And then the next study is about urinary extracellular vesicles and cell-free RNA. So again, we collected urine and treat them with urine preserve and store at room temperature for up to 7 days and compare with unpreserved samples. So we, just as Nick mentioned before, we use nanoparticle tracking analysis, the nanosite, to quantify the number of extracellular vesicles. So we use a precipitation method to isolate them. And then, as you can see in the figure on the left, in the urine preserved samples, we maintain both the count and the size of the isolated urinary extracellular vesicles. And then further on, we we extract the total RNA from the isolated UEVs using the QIAGEN kit with adaptive protocol, and we measure housekeeping messenger RNA, beta-actin, and GAPDH. Again, using ddPCR, as you can see from the figure on the right. For both genes, the unpreserved sample, even at day 3, barely any of the messenger RNA were detectable. And then with the Streck urine preserved sample, they're maintained fairly well over the storage of 7 days. And then the last study I'm going to share with you is to show that our urine preserve can maintain the integrity of the exfoliated cells. So in this study, we collected urine from healthy donors and we spike in the human breast cancer cells that are stably expressing green fluorescent proteins to mimic the cells that shed into the urine. So we treat these samples with the urine preserve and store at room temperature for up to 7 days and compare with unpreserved samples in parallel. So, and then we immobilize the cells onto the slides, and then the slides are further stained with hematology stains and imaged under the microscope. So here you can see from the figure on the right, so the unpreserved samples, from day 4, the cells start to degrade, and then day 7, all the samples, the only thing you can find is debris. But if you're looking at the Streck urine preserved samples, all the cells remain intact. And then we also have another set of these sample slides and look at them under the fluorescent microscope. And we use DAPI to stain their nuclei. And again, very consistent with the previous result. In the unpreserved samples, we barely see any green fluorescent cells there, starting like day 4. And then in the preserved samples, the fluorescent intensity are pretty strong and the cell remain intact. So the take-home message for the urine part is for urine liquid biopsy, the urological diseases, urine can actually prove to be liquid gold. Currently, there are some assays that are developed for kidney injuries or non-urological cancers, but they're still at very early stages. And then urine is a very unique biofluid, so it can be collected anywhere by anyone. And also keep in mind, urine is also a very harsh environment. Because of these molecular diagnostic tests, the downstream extraction and analysis for these urinary targets can be expensive, time-consuming, and difficult. And we know that up to almost 70% of the laboratory errors is related to pre-analytical variations. So we're hoping Streck urine preserve is the solution that can help minimize the pre-analytical variations to avoid the cold chain transport and maintain the cellular and cell-free targets. And you have trust in the integrity of your sample. And then I'll turn to Nick to conclude this webinar.
Okay, so just 1 or 2 last slides in conclusion. So liquid biopsy, utilizing either blood or, as Jing talked about, urine matrices, is fast becoming routine in the clinic as a means of monitoring health and or disease status. And we know for both blood as well as urine, sample stabilization is critical. In both cases, analytes are generally, if not always, found at extremely low concentrations. So again, back to the needle in the haystack analogy, it's already this hard to identify the needle in the haystack. Any kind of extra degradation from red cells in a blood sample or white cells in a blood sample, et cetera, just makes it that much harder to identify your analyte of interest. So Streck offers solutions for sample stabilization. multiple offerings for whole blood, and then the single urine preserve offering for urine. And then lastly, if you plan to use any of these stabilization products in assay development, please contact the distributor so that we can lend help if necessary in development of your assay. And with that, thank you very much for attending this webinar.